Ever stop to think about how we get bread to rise, or how something as complex as insulin is made? Well, the answer is all around us, in a science that connects our kitchens to cutting-edge hospitals. So today, we're going to dive into the absolutely incredible world of biotechnology. Okay, let's kick things off with a bit of a riddle. What on earth could bread, cheese, and modern vaccines possibly have in common? I mean, they seem totally different, right? You've got your food, your dairy, and then a medical marvel. But believe it or not, there is one powerful idea that ties them all together. Any ideas?
The answer is biotechnology. In the simplest terms, it's just us using living things, like the yeast in bread or the bacteria in cheese, to make products that make our lives better. But here's the key thing. Humans have been doing this for centuries, but what's happening now with modern science has completely supercharged what's possible.
To really get a handle on modern biotech, we've got to pop the hood and look at the engine. And that engine is a process called genetic engineering. This is the technique that has changed everything, and it's a core concept you absolutely need to nail down. So let me introduce you to the star of our show, the humble bacterium. Now, scientists just love working with bacteria. Why? Because they're simple and they grow incredibly fast. Inside, you see its main chromosome, right? But the really important part for us is that little separate ring of DNA. That's called a plasmid. For any exam, you have to remember this word. The plasmid is the absolute key. Just think of it like a tiny programmable USB stick for DNA. So what's the grand plan here? What's the goal? It's actually really elegant. We want to take one of these normal, everyday bacteria and reprogram it, turn it into a tiny living factory. And this factory isn't making cars or phones. Nope. It's programmed to turn out one specific, really valuable product, like the insulin that people with diabetes need. So how in the world do we do that? Well, it's basically a biological version of cut and paste. First, you find the gene you want. Let's stick with the human gene for making insulin. Then you use these amazing things called enzymes. They're like microscopic scissors to cut that gene out. Now, here's the really clever part. You use the exact same enzyme, the same pair of scissors, to cut open that plasmid we talked about. Because you use the same tool, the cut ends are a perfect match. They're kind of sticky. This lets us paste the human insulin gene right into that bacterial plasmid. And this new combination piece of DNA, it gets a special name that you definitely need to know. Recombinant DNA. Makes sense, right? It's been recombined from two different sources, our human gene and the bacterial plasmid. Then, the final step is to take this new recombinant DNA and pop it back into a bacterium. And the result? Presto, you've got yourself a genetically modified, or GM, bacterium. This isn't just a regular bacterium anymore. It's our tiny factory. And here's where the magic really happens. Bacteria multiply fast, so that one single GM bacterium soon becomes millions, then billions, and every single one of them is pumping out human insulin. All we have to do is harvest it, purify it, and there you have it, life-saving medicine. Okay, so we figured out how to build these amazing little factories. Now let's see what they're doing out in the real world. Because beyond medicine, one of the biggest places biotech has completely changed the game is in our food and how we farm. Just take a look at these two pictures. It's a perfect example of what we're talking about. The image on the right shows a high-yield crop. This is biotechnology in action. These plants have literally been engineered to be stronger, healthier, and to produce way more food using the exact same amount of land. It's a total revolution for agriculture. So how is this even possible? Well, scientists use that same genetic engineering toolbox to give plants all sorts of new abilities. It's like giving them superpowers. They can boost the yield, make the plants resistant to annoying weeds and pests, help them survive a drought or in salty soil, and even help them fight off diseases all on their own. This just makes farming so much more efficient and helps us feed a growing planet. A really powerful case study for this is a crop called golden rice. You can see why it gets its name. It was engineered not just to produce more food, but to produce better food. It was designed to solve a massive global health crisis. And that crisis is vitamin A deficiency. In many parts of the world, rice is the main thing people eat, but it doesn't have any vitamin A. This deficiency is a huge problem, especially for kids, and can lead to blindness and a weaker immune system. So what scientists did was engineered golden rice to produce beta-carotene. That's the stuff in carrots, which our bodies then turn into vitamin A. It's an incredible example of using biotech as a direct tool to improve public health. And that leads us perfectly into our next big area, how biotechnology is being used directly to improve human health on the medical frontier. So, using that exact same bacterial factory method we've been talking about, scientists can now make a whole pharmacy of life-saving products. We're talking huge amounts of pure insulin for people with diabetes, powerful vaccines for diseases like hepatitis, growth hormone, even natural painkillers like beta-endorphin, and things called interferons, which are proteins our own bodies use to fight viruses. But here's the thing that's truly revolutionary, the part that really gives you chills. Biotechnology isn't just about making better versions of old medicines. It's about creating hope where there was none. It's being used to develop treatments for diseases that, not too long ago, were considered a death sentence. It has completely changed what's possible in medicine. Okay, I know that was a ton of information. So let's pause and do a lightning round recap to lock in the most important points. If you've got an exam coming up, this is the part you really want to pay attention to. All right, let's nail down these definitions. Ready? Biotechnology, simply using living things to make useful stuff for us. Genes, they're the specific instructions in DNA for building a protein. And genetic engineering, that's the technique of moving a gene from one organism into another. Got it? Awesome. Next, the big concepts to remember. Recombinant DNA, that's our cut and paste hybrid of a gene and a plasmid. The GM bacterium, that's our tiny factory that's running on that new recombinant DNA. And its applications? They're everywhere, from farming and health to even cleaning up environmental pollution. These are the core ideas. So let's end with this one final thought. We now have this incredible, almost unbelievable power to edit the code of life itself. We can design plants to fight hunger and program bacteria to cure diseases. So the question I want to leave you with is this: what's next? What is the next great challenge that our generation should solve using the power of biotechnology? It's a lot to think about. Thanks so much for tuning in.